Hello, everyone. So my name is Arutha Kulasinghe. Thank you for the introduction, and I'd like to first of all thank Cure Cancer for the funding that's enabled this work and really supporting early career researchers during this difficult or challenging stage. So my topic that's been funded by Cure Cancer is the spatial profiling of the tumor microenvironment. So I'll just dig a bit deeper into that and talk about the research. So this work was first described by Jim Allison, and this was a photo taken at AACR last year, where he showcased some of and he talked about the pathway to getting the CTLA-4 work up, and immunotherapies have done really well in a certain subset of patients. The work's really led from melanoma, but it's shown like real durability in non-small cell lung cancer, head and neck, bladder. So in a subset of patients, they tend to do really well with immune checkpoint inhibitors, and identifying those subsets of patients is an unmet clinical need, and that's what my research is hoping to do, to look at the tumor microenvironment, understand it at a greater depth, and see if we could predict a response to checkpoint inhibitors. So as I mentioned, one way to do this is to look at the tumor microenvironment, detail it at a greater depth than has been previously possible. Another sort of way to do it is to look at the mutational load or the tumor mutation burden. So if you look at this graph on the right here, a higher mutational load associates with a better outcome. You've got a higher neoantigen load. And this is shown to be tissue agnostic. So last year there was some data that came out that showed in patients with a high mutational burden, they tended to do better with immune checkpoint inhibitors. And so this just came out recently, where Merck approved Keytruda for high TMB patients. Anyone with a TMB of more than 10 mutations per megabase was given this therapy. So it was tissue agnostic, and then this is one of the first times in cancer where it hasn't been tissue specific. So one of the challenges in looking at the tumor microenvironment is on this, on the bottom right here, you've got two images here, and these are two tissues which have similar inflammation signatures based on bulk expression data. And on the left, what you can see is that the green is where the tumor regions are, but you don't have a real immune cell infiltration into that. But on the right, you do. So both these tumors have similar inflammation signatures. But when you look at them with the spatial context and actually see where the immune cells are, whether they're recognizing the tumor, the difference there is on the right, you've actually got infiltration into the tissue, and in that tumor, those tend to do better with immune checkpoint inhibitors, whereas the tissue on the left is excluded from the immune cells excluded from the tissue, the tumor regions. So one way to do this is to digitally spatially profile this, and what this technology allows you to do is it's antibodies bound to UV photocleavable linkers, and when a UV light is shone above these regions of interest, it liberates the signal from that region. So you actually get a digital count at the RNA level. You can do about 1800 plex RNA, or at the protein level, about 100 markers spatially. And you do get a whole slide overview. So you're indirectly, without destroying the tissue, you're able to really profile the tumor microenvironment at a greater depth than has been previously possible. So this technology is called nanostring DSP, and it's a really exciting technology that we've recently brought to QUT. So this is some initial data where on the top you've got the H&E sections from this from a number of head and neck patients. You've got a mutational burden for one of the patients where there wasn't sufficient tissue to do the whole exome, but there was for one case. In the middle, what you see is this is the nanostring whole tissue staining of the sections, and then you're able to go into different regions of that tissue to plex to look at a higher plex. So you typically do 12 or 24 regions within that tissue and try to get a representative area in the tumor, the microenvironment, stroma, and the normal regions, and then extract the data from there. And what we found in this initial study was that the dogma or the literature in the field showed that CD8 T cells infiltrating the tumor was predictive of outcome or a more favorable prognosis to immune checkpoint inhibitors. In this head and neck cohort, though, what we found was that when we looked at non-progressive versus progressive disease, CD8, or those immune or those T cells weren't predictive of outcome to therapy. So this is at the protein level, and just running across a number of, this panel was about 60 immuno-oncology targets. But what we found was that there were a number of targets that were really interesting, and this wasn't seen before in the field. So CD44, STING, CD66B, phospho-AKT, and PTEN did associate with a more progressive disease profile. And now what we're trying to do is go back and do whole slides assessments at about 7 to 8 plex of these markers to see if we can understand it at a greater depth. So it's giving us insights into the tissue biology that we weren't able to look at before. And I'll quickly just give you a snapshot of lung cancer. This is a tissue microarray. So you've got about 90 lung cancer cases with matched normal. And what we did was we wanted to use a geomasking function. What this allows you to do is to draw around the tumor regions, liberate the signal from the tumor regions, and you get an actual digital count for the tissue, for the tumor regions. And when you do the inverse, you get the complement of that, or the immune or the microenvironmental signature. So you get 2 signatures per core. And that's really exciting for us because we're able to profile that entire core by simply just drawing around it with a pathologist. And what we're able to see, without going too much into the data, is on the right here is a survival analysis for some of the patients. So what we found was that CD34 and CD3 seem to be protective in these tumors, and granzyme B, fibronectin, and a number of other markers seem to associate with a worse outcome. So this is very preliminary data. It was probably collected in the last few weeks, but it's really exciting to see these markers that weren't previously possible to be seen at this depth. And these are some of the assays we're hoping to run in the next 6 months. So these you're able to do about 18,000 mRNA targets spatially again. So you're able to look at the signaling from the tumor regions into the into the microenvironment. So in the bottom left here, you can actually see how the signaling changes. So interferon gamma and a number of different pathways. You're actually able to map that within the tissue context. And that's really exciting. So this is what we're hoping to do in the next few months. So it's really, really exciting work, and it's at the forefront of spatial biology, and we're really encouraged by the initial data set. With that, I'd like to thank the funders, Cure Cancer and Can2. This was a photo taken at earlier this year in Sydney, and it was really great to meet the alumni, the funding agencies, and the whole team at Cure Cancer. And I'd just like to say that since this meeting, we've really begun collaborating. So Fernando and I have collaborated quite a bit, and I'm hoping to extend that work on with Mark Adams. So a lot of Cure Cancer alumni have come together from just attending these meetings. So we probably sit in the same building, but we had to go to Sydney to actually meet each other, which was a coincidence, but it's resulted in a lot of exciting collaborations. Thank you.